Okay, hello everyone. Thank you, Eglantine, and for that introduction, and thank you to you and the organizers for inviting me to be here with you all today to share a little bit about our research projects. I've really enjoyed the intellectual exchange on this RTB Foods webinar platform over the last year, and so I'm really looking forward to our discussion today. So I'm a research food technologist with the USDA and the Agricultural Research Service, which is the chief scientific in-house research agency of the USDA. And we have more than 600 projects and 2,000 scientists and that represent the diversity of expertise that you can imagine associated with agriculture. And our lab is located here in Raleigh, North Carolina on the East Coast. And the majority of my projects contribute to one of the 15 national programs, the one focused on product quality and new uses. And the mission of that national program is to conduct research towards developing more healthful value-added foods, especially through reducing loss and waste using commercially preferred technologies for post-harvest processing, packaging, and storage. Everywhere you see a red state indicates a state that houses a research program associated with product quality and new uses. And ours is located in Raleigh, North Carolina, and I'm one of 5 PIs in the Food Science and Market Quality and Handling Research Unit. And my program is focused in on vegetable preservation, composition, and quality. Specifically, our long-term goal is to develop science-based technologies that enable sustainable preservation of fruits and vegetables, resulting in high-quality, health-promoting consumer products. And so we work in a variety of research areas. A lot of work that I've done in the past has been related to understanding the biochemical changes during lactic acid fermentation. We've developed some analytical methods for metabolite profiling to get a broader view of the composition of fruits and vegetables. We work independently and collaboratively on different processing technologies, and with a big focus on product quality and healthfulness, in incorporating techniques like texture analysis or sensory analysis. And I've had the great fortune during my time as a scientist with the USDA to be co-located at North Carolina State University and be able to mentor more than 60 students, I think now, graduates and undergraduates, a mix of graduate and undergraduate students in research associated with fruit and vegetable preservation. And so in general, in my lab, we use a 3-pronged approach to try to get a better understanding of the chemical composition of fruits and vegetables that relates to product quality. And we do that by adapting or developing analytical chemistry techniques like GC mass spec and LC mass spec or other HPLC techniques, and combining that with information from some kind of routine product quality tests like texture analysis and color imagery, as well as a variety of different sensory analysis methods to give us the sensory information we need to meet whatever project objective that we're doing. So among the commodities that I've worked on, primarily pickled vegetables. More recently, I've worked on several projects related to sweet potato, and that's how I came to know this RTB Foods group. And it's been really a great interaction, and I'm happy to continue working together with this group. So of particular interest is the sweet potato among those commodities. And for us in the US, one of the reasons for this is that fruits and vegetables are recommended for improving our overall health outcomes over the course of our lifespan. And in particular in preventing chronic diseases that kind of plague us here in America. We don't have nutritional deficiencies so much as we do have imbalanced dietary habits. And in fact, more than 90% of our population is currently below the recommended intake of red and orange vegetables. So sweet potato is really uniquely positioned to fill this need in our diets, as it can produce more nutrient-rich biomass than any other food crop worldwide. It's highly nutritious, as I think all of you are very familiar with. And there's a high degree of biodiversity in the germplasm. So this means that we have a lot of opportunities to develop unique, novel value-added food products to increase consumption and utilization of this vegetable. And so this increased utilization and consumption of sweet potato is achieved through integrated advances in breeding, crop management, and food science, to develop new varieties, develop new processing methods, and ultimately all of these things have to meet consumer preferences or people still are not going to eat it. And so that's where we've partnered with the breeding and crop production programs in the Department of Horticulture at North Carolina State University, and more recently with the International Potato Center, which is our connection to this group here. And so we've known for a long time that the cooking process really influences the eating quality of the sweet potatoes, and that this is key to getting people to consume more sweet potatoes, kind of as shown here by an old, more than 100 years old publication from the USDA that was put out for farmers on how to best prepare their sweet potatoes for consumption. For full flavor development. This is important because the long times that it requires to prepare sweet potatoes is also one of the things that actually prevents more consumption of sweet potato, especially in kind of the fast-paced world that we live in over here. People don't always want to take that time to be at home cooking. So some of our early projects where I collaborated with Dr. Van den Trong were on actually looking at how can we come up with products that are more quickly prepared and can be, increase that consumption by having something that's either quickly prepared or ready to eat. And so one of the interesting projects we worked on was actually to try to develop a fresh cut sweet potato dice in microwavable pouches. And so for those of you that aren't familiar, we have here, most households have a microwave oven, household sized microwave oven, looks like a box, and it's using electromagnetic energy to heat the food rather than the traditional heat sources. And it's very rapid. So it's a very rapid cooking process. So we developed these fresh cut sweet potato dices, packaged them in a microwavable pouch, optimized the microwave cooking protocol to give us a product that looked very similar to possibly a boiled sweet potato product here in the US. And then one of the things with a product like this for the market is that we need to do some sort of rinsing protocol whenever you have a fresh cut produce. Most of those commercial operations are using a rinsing treatment to prevent microbial growth. And so basically, we found that the typical chlorine rinse treatments that are used in a lot of commercial fresh cut processing operations here resulted in a significant off aroma when combined with the sweet potato. And so alternative sanitizers would need to be used, but those alternative sanitizers did give a very similar appearance and flavor to the freshly prepared dices when we used a difference from control test. In another study then along those same lines of trying to kind of speed up the cooking process for sweet potatoes, evaluated a couple varieties to see if the variety could, if the Evangeline variety could perform better as a microwaved sweet potato compared to oven baking. It was thought that this might be the case because this variety was developed with a higher sugar content in mind. And so this was a study, again, in collaboration with our partners in the Department of Horticulture, who grew these Evangeline and Covington both in North Carolina. And then we evaluated their property, their chemistry, and also their sensory properties using a consumer panel, using a hedonic scale for liking. And what we found was that generally these two varieties were equally well liked, although the Evangeline was more well liked for this deep orange color that you see. However, the microwave sweet potatoes, although we spent a lot of time in the pilot plant optimizing our cooking protocol for that, the just about right scales showed that the microwave sweet potatoes were still less well liked due to lack of moistness, flavor, and sweetness. And so then looking a little more deeply into that, we see that basically both of the varieties were similar in liking here, except for color, where Evangeline stood out with its deep orange color, which was preferred with this particular consumer population. And then, but the big differences were seen in how the sweet potatoes were prepared. And we saw that this played out mostly in the texture and flavor attributes, resulting in the microwave sweet potatoes being less well liked overall. We see then when we look into the chemical composition of those same sweet potatoes that were presented to our consumers, that lack of moistness that was indicated in our just about right scales is shown out here by more drying happening during that particular cooking protocol that we were using in the microwave, and that even this small difference in moisture is something that influences the consumer liking pretty significantly. And as we might expect, that difference in lack of sweetness was due to basically a lesser maltose production in the microwave sweet potato. Looking into other ways of how we can increase the consumption of sweet potato here in the US is to look at products that are ready to eat and that are popularly consumed. And so fried products like French fries and chips have been a main focus of our work as well. And what I'm showing you here is a principal component analysis of a texture study done with a variety of sweet potato cultivars to evaluate their textural properties for developing new varieties that would be better suited to commercial French fry processing. We see that our most commonly produced ones here in North Carolina, Covington and Beauregard, are on the softer side and more cohesive and smooth on the inner portion of the fries, moist and smooth inner portions, whereas the commercial sweet potato French fry, which is coated in order to retain its textural properties, is grouping over here with some other cultivars that may have potential for development as a variety specially suited for French fry processing. We looked into the chemistries, and as in other studies, the overall hardness, or this is sometimes called firmness, was certainly related to dry matter content of the raw sweet potato. But this important attribute of denseness, which is how compact the cross section is perceived, is a very important attribute for characterizing sweet potato French fries, and is kind of opposing to commercial white potato French fries, which are hugely popular here and eaten just about everywhere in the US, which are very light and airy on the inside. So versus this very dense or this light and airy. So this is an important property for us to better understand. So we looked more deeply into the starch properties of these same sweet potatoes that were characterized for their sensory properties, and isolated the starch and did pasting profiles and also texture profile analysis of the resulting gels and pastes. And what we found that was interesting was that this French fry denseness, which could not be explained by any of the other measured properties like dry matter, starch alone, was correlated with the pasting profiles of the starches from the different French fries, as well as the non-starch alcohol insoluble solids component of the French fries, but not the raw sweet potatoes. So I think there's some interesting things here for further exploration into those individual starch characteristics and also the non-starch components of alcohol insoluble solids that are contributing to these interesting textures that really drive consumer preferences well beyond just the overall hardness or firmness of the product. So as we use frying as a technique to increase the utilization and consumption of sweet potato, our goal is to get healthy foods into more humans. And so we also need to make sure that we're paying attention to the chemistries that might be undesirable in a fried product, and most notably is acrylamide formation in fried foods. This is true of all fried foods, not just sweet potatoes. And so we did a project on sweet potato chips looking at acrylamide mitigation through varietal selection. And what we see here is that among these different cultivars that we tested, there's a very varied profile of free amino acids in these sweet potatoes, as well as sugar contents and compositions that can contribute to acrylamide formation. And in looking into this further, then we constructed a model to determine which of those amino acids and sugars were playing the greatest role. And of course, as some of you who are familiar with acrylamide might know, asparagine and glucose are known as kind of the main precursors to that. But for the model to be really predictive of the final acrylamide content in sweet potato, we actually needed to include several other free amino acids and sugars in the model, as they may be competing or contributing in different ways to those chemical reactions in the final acrylamide content. I think most interesting here is that that things are happening differently in sweet potato chips than they do in white potato chips. There's been a huge amount of work on white potato or the Irish potato chips in terms of acrylamide mitigation. And in those systems, the browning is often related to the acrylamide content. And so they use browning as kind of an easy indicator of acrylamide content. However, in sweet potato, this browning has no correlation whatsoever to acrylamide content. So we cannot use browning index as an indicator. We need more sophisticated techniques, and there's multiple components that influence the level formed. And so I think that there's more for us to learn here, and we can use this kind of information to screen for varieties that will form lower levels of acrylamide during frying. And that brings me to my time here with all of you in RTB Foods. And this is a photograph from 2019, the work that Mariam and Lise and I did together at Narrow in 2019 to initiate the development of an international sensory lexicon for sweet potato. We call this phase 1 Uganda, because we do intend to expand this work to the group in Mozambique, as well as utilize it here, adapt and utilize it here in the US. And so what we did in a very intensive week together was we presented a wide variety of sweet potato cultivars to our team of sensory panelists there, and had them describe in their own words all of their characteristics: aroma, appearance, flavor, and texture. And then through a series of activities and discussions, we basically narrowed that down to 36 descriptive terms for sweet potato. And we looked to the scientific sensory literature to, I guess, interpret colloquial terms into standardized sensory terms that could be used universally across groups. And then we went to the market and we looked for reference foods that would be commonly available there that could be presented to the panelists to help train them on the definitions of these attributes and give a reference material that could be presented frequently as help in training so that people use the scales consistently. And then we practiced, we developed some draft scales and practiced with the panel and worked together to develop this draft sensory analysis workflow for sweet potato samples. And what you'll notice here is that there's a number of stages. So one of the things for consistency and for generating good sensory data is that you need to also standardize the method of evaluation for all of these attributes. And so this was our first go at standardizing the method of evaluation. And I know that Miriam has continued to refine this with her panel, and I'm sure we'll be hearing more from her on this in future RTV Foods and Sweet Gains activities. And so something I do want to point out, though, was just kind of interesting about sweet potato is that we have all of these aroma, appearance, flavor, and texture. And what you'll see is a large number of those attributes and the different ways that we need to evaluate them has to do with the texture of the product. And it's just so fascinating. Sweet potatoes are just incredibly diverse in the textures they can produce. And I think there's a lot of interesting things that we can work on in the future in this arena. And so our current project is really focused in our current USDA project. We have several things that we're working on, and in sweet potato, our focus is in looking at linking the raw sweet potato composition of different cultivars under different processing conditions with those sensory properties in order to determine the underlying physical and chemical characteristics that we can use to develop novel value-added products that are going to be what consumers are looking for. And we have a full-time postdoc on sweet potato processing and chemistry, Dr. Matt Allen, who will be with us for another year. And his focus is on the sweet potato polymer structures and texture. And newly onboarded with us, Ms. Lisa LaFountain, who was previously a student with me, and some of you got to meet when we came to Uganda in 2018. We were able to hire her as a permanent support scientist in March, so I'm so pleased to have her back with me, and she'll be splitting her time between pickled vegetable research and sweet potato work with a focus on helping with the descriptive sensory analysis panels. And specifically, we want to look at how volatile compound compositions can play into the perceived sweetness of sweet potatoes. And with that, I definitely want to acknowledge all of the sensory panelists over all the years and consumers that have participated, the North Carolina Sweet Potato Commission and other industrial funding that we've had through our collaboration with North Carolina State University, the International Potato Center, and these RTV Foods and Sweet Gains projects, all the collaborators and co-authors, a few of whom are shown here, and I'm looking forward to working with all of you and adding your pictures to this slide someday. And with that, I'll open for discussion.
Thank you very much, Suzanne, for these very clear, very well illustrated and lively presentations, which also highlights the remaining challenges that have to be handled within projects such as Sweet Gains and RTB Foods. So we have lots of sweet potato experts today in the room, from SIP, Naro, Uganda, GHI, Scotland. So I'm sure that you have lots of questions in your mind, so don't keep these questions for yourselves and share with Suzanne and with the group. First of all, there are questions from Dominique. How did you evaluate the sweet potato preferences in the USA at an industry level or at home level for homemade products?
I'm sorry, can you repeat the question?
How did you evaluate the sweet potato preferences at industry level or at home level for homemade products? Were you working with the industry or
yes,
with...
so the consumer the consumer panel, we prepared those products in our pilot plant facility, and then we recruited consumers from the public to evaluate the liking of those products. So I think we had about 150 sweet potato consumers come to our facility and evaluate the products.
I think that answers the question. Thank you. Dominique is also mentioning that orange sweet potato is popular in the United States, but what about white and yellow genotypes in the USA?
Yes, we don't see those in the common marketplace yet. I think historically the very sweet, soft varieties are what our consumers are used to, but there is a growing interest in novel foods in the US. There's the younger generations are interested in things that are different. So I think there's an opportunity for those to come into our market, but we have not seen them. We don't see them commonly. There are some white or cream fleshed, but you have to go to a specialty grocery store to get them.
Okay. Do you have any idea why density or denseness is related and opposite to amylose content?
No, but I will say that the amylose content, that's where it came out on the map with the PLS regression model, but there was not much variation in amylose. There was not much correlation of amylose content with any of the textural properties in the French fries.
Okay. Dominique, is that okay for you, for your questions?
Yes, yes. I think you have a very small diversity. You are working on some few clones, not on a panel of diversity.
Perhaps for the amylose content. These cultivars were selected to range in dry matter content. So there was a wide range of dry matter content, and it did include orange, yellow, and cream fleshed and white fleshed sweet potato for the fries. But we were not specifically selecting for a range of amylose content. That's correct.
Yes, thank you, because normally it's linked to hardness and density, and really. Thank you.
A question from Emmanuel from IATA. What other components could you suggest that contribute to the non-enzymatic browning in sweet potato? Components contributing to non-enzymatic browning.
So I think the components, the amino acids and the reducing sugars are the primary components there. We did not specifically model browning with our amino acid data, but I think that could be interesting to do.
Is that okay for you, Emmanuel? Or do you want to ask something more? Complementary information to Suzanne?
Yeah, it's okay. It's a good study as she has mentioned to look at, and that's what we want to look at also in RTP foods. So we want to look at other components that cause this non-enzymatic browning, apart from the Maillard reactions, reducing sugars presence. Thank you.
Thank you. A question from Michael. How about oil absorption, greasiness of the fries? How does that clash or not with the texture quality and the biochemical composition?
So the oil absorption was inversely related to the dry matter content in this study. So with higher dry matter content, we had less oil absorption, and that was somewhat related to that perception of surface oiliness on the fries.
Michael?
Thanks, Suzanne. Sorry, I kind of came in a bit late from another meeting. So in a way, that's, as we're looking for high dry matter content, it's good that there's no clash there.
Yes, and at least in our study, then you would have less. We would expect that there would be less oil absorption.
Dominique is asking, are you working on milliness and parietal compounds?
Milliness and what?
And parietal compounds. Dominique?
Cell wall structure and milliness. Is it something
I don't know the term. What do you mean by parietal?
So is it important in the USA, the milliness of the sweet potato for the people, for the consumption, or not? Is the milliness something that you are looking at?
It has not come out as an important sensory attribute here for the traditional orange flesh sweet potatoes. They tend to not be mealy. So it has not been something that we've been focused on, although I do think that we're going to have an opportunity to work on that with our partners in Mozambique as part of the Sweet Gains project.
Okay, good. We are together.
Yes, this is a great group.
Yes. Also about the maltose, is it an issue in the US, the production of maltose during cooking? Because some clones are producing a lot of maltose, other clones they are not producing. Is it an issue?
Our research is showing that it's a desirable trait. and I know that our the breeding group that we work with, Craig Inshaw's group, they have varieties that have a wide range of amylase activities, and we're planning to look at those in terms of sweetness development. They've done some work already on that, I think. They're trying to link it using the genetic mapping techniques, but I think that's still a work in progress. But maltose development, I think one thing that we have in common between the consumers in the US and the consumers in Africa is that everybody likes the sweetness. So I think that the maltose formation is probably the main driver of that. But we may also have some influence of the volatile compounds on the perceived sweetness. So that's what we hope to be looking more deeply into.
Linked to the previous question from Dominique, there's a question from Mark. Have you looked at cell wall composition in relation to textural properties?
Not yet. That is work in progress. That is what our postdoc, Dr. Matt Allen, is looking into that, along with the starch characteristics, individual starch structures. But as with everyone, we're delayed. He started right when we went into quarantine, so he's had mostly time to do theoretical work and not as much time in the lab. But we're hoping to get more deeply into that in the coming year. I will say on that we have we've experienced some unique genotypes where they become rubbery, and instead of fracturing normally, and we do think that that is related to the non-starch polysaccharides, but we haven't had a chance yet to look into those structures.
Thank you. Mark, do you want to react directly?
Thank you for that answer. It's something that we're quite interested in ourselves, and we're awaiting some results. So interesting to compare how we get on with that. You know, we're looking at pectin methylation as well as composition of the cell wall polymers in samples that have been evaluated for texture. So there might be some synergies we could develop.
That's great. I would welcome the opportunity to talk more deeply with you about that offline.
Yeah, let's do that sometime. We'll fix up a meeting at some point. Thank you.
Great.
Arnold from Naro, Uganda is asking, you evaluated sweet potatoes under storage for a period of 14 days. Then what was the level of MDA equivalent as the major biomarker for lipid peroxidation? So level of MDA.
We did not look at that. In that study, we were only looking at, we looked at the microbial counts for preservation and the sensory properties, but we did not look into the chemical changes during that time.
Mm-hmm. Okay. Any other question or comments from the sweet potato expert? I mentioned SIP, I mentioned GHI, Naro, but there are also people from CNRA Côte d'Ivoire working on sweet potato.
Andrade from Mozambique also.
SIP Mozambique as well. anyone who would like to ask for clarifications or make suggestions?
Oh, I see a direct message to me. I think the question is, to what extent are you using stereochemical analytical approaches to evaluate stereo sugar effects on acrylamide formation? And then a follow-up, don't you think non-targeted metabolomic approaches can be adopted in this kind of work, especially on effects of glycoamino interactions and flavor chemistry in sweet potatoes? And so the answer is, we have not looked at the stereochemical interactions. And I do agree that non-targeted approaches can be adopted. And we hope that we can get more funding in the future to be able to incorporate that. We've developed some non-targeted approaches in our lab for volatile compounds and also for the metabolites of central metabolism. But as of now, we need more funding to be able to apply that to sweet potato.
Good, thank you. Okay, question from Yusuf. Don't you think non-targeted metabolic approaches can be adopted in this kind of work, especially on effects of glycoamino interactions and flavor chemistry in sweet potato? So non-targeted metabolomic approaches. Do you have any comments on this experience?
Yes, so I do think those can be adopted, and we hope to get more funding in the future to be able to apply those. For the, I guess for the flavor chemistry part, that is part of what we're planning to do with the volatile compound profiles and sweetness perception. So we have a 2D gas chromatography mass spectrometry system, and we're planning to do non-targeted profiling of the volatile compounds in relation to sweetness perception and hopefully other flavors as well.
Another question from Yusuf in the chat box. To what extent are you using stereochemical analytical approaches to evaluate effect of stereo sugar effects? To evaluate effect, well, on acrylamide formation? Oh, that's even too complicated for me.
Yeah, I think.
You'd like to answer the question?
Oh, I'm sorry. I think we answered that question, but I can just restate that we have not used the stereochemical approaches.
Yeah. Good question from Maria Andrade. What is your final recommendation on cooking sweet potato to remain the flavor?
Oh, so that's a good question, Maria. So I think that ultimately that will depend on the consumer group that you are targeting for whatever product you're developing. So for our traditional sweet potato products here in the US, probably baking or steaming or roasting. The frying, actually, even in the frying, the flavors do seem to develop. We get characteristic sweet potato flavors in those French fries as well. So I think it's going to depend on your target group of consumers, which is why even with the descriptive sensory analysis panels, if we can come up with an international lexicon that we all use together, we'll still need to link that to the preferences of target markets for different products and possibly different varieties.
Michael is also mentioning something that in Africa, in North America, consumers usually prefers very thin pieces of sweet potato fries, whereas in East Africa, maybe it looks like consumers prefer much larger pieces. So it should be confirmed by East African colleagues, of course, but could this affect the sensory attributes?
Yes, I think most definitely. I think that it would depend on the frying method, but the heat transfer that you get during frying would certainly be affected by the sample geometry.
Sure. And Arnold is making another comment. He thinks that regarding the storability work, it's important to determine the effects of reactive oxygen species, because you may be actually dealing with stress-related factors. That was a comment more than a question.
Okay, thank you for that comment.
Any last question or comments? There were a lot of questions, actually, reactions. No? Okay. So I'd like to thank you very much, Suzanne, for this presentation of your work at USDA, which I'm sure will pave the way for future opportunities for collaborations within RTB Foods, within Sweet Gains, and so on. So thanks a lot. Looking forward to more opportunities for working together or meeting, even virtually. So thanks a lot. I'd like to let you know that in 2 weeks' time, there will be no webinar. So on July 2nd, there will be no webinar, and our next webinar will be on July 16th. And this should be a major webinar because we should have a presentation from the Work Package 1 coordination team on the methodology to consolidate the RTB product profiles. So how to extract key information and knowledge generated through surveys and consolidate a product profile that can be used by Work Package 2 partners, food scientists, and at the end by breeders, Work Package 4 partners within RTB Foods Framework. So the whole methodology should be presented on July 16th. So I hope you will be able to attend this presentation. And then after July 16th, there will be no webinar for a month's time because the PMU will be on leaves, on summer leaves. So last webinar before summer leaves will be on July 16th. So thanks everybody. Thanks a lot, Suzanne, and take care. And we see each other in a month's time. So July 16th. Thanks everybody.
Thanks, Suzanne.
Bye.
Thanks, Suzanne. Thanks,
Thanks everyone.
Take care.
Bye.
Thank
Bye,
you.
Mark.
In Uganda. Take care in Uganda for the new wave of COVID.
Bye, everyone.